Bismillah ar-Rahman ar-Rahim. This is a description of the nervous tissue data show. This picture demonstrates the neuron or the pericardium, which is the cell body. It is named the pericardium because it surrounds the cardium, which is the nucleus. Here you can see that the body of this nerve cell is showing processes, and it is mostly a multipolar, as we have many dendrites as well as the axon. Here you can see that the nucleus is pale with evident nucleolus, and the cytoplasm is full of the basophilic granules called Nessel granules. These Nessel granules are the free ribosomes in the focal type of arrangement, and also you can see these Nessel granules present in this part. So this part is a dendrite because the Nessel granules are absent from the axon. On the right side you can see these multipolar nerve cells with the vesicular achromatic nucleus with prominent nucleolus, and the Nessel granules filling the cytoplasm of the cell body. Here is the same, but in these areas you can see a lot of cells. These cells are the neuroglial cells, which are in number 10 times more than the number of the neurons. They are supportive in function, and the most common type is the astrocytes. And as these astrocytes are present in between the cell bodies, it is the cytoplasmic astrocyte. Don't forget that the Nessel granules are the first to disappear during the degeneration, and this process is called chromatolysis. Chroma means the color because these granules, like the basic stain, and they are stained, they have a color. So the chromatolysis is the one or the first sign of degeneration, and it is a disappearance of the Nessel granules. The Nessel granules amount reflects the metabolic activity of the cell. This picture demonstrates the cell body or the pericardium of the neuron, together with other cells that are the neuroglial cells. And you see that the neuroglial cells are more than the nerve cells. Actually, they are 10 times more than the nerve cell number. Here we have the nucleus, and this is the cytoplasm. The cell body is called the pericardium as it surrounds this nucleus, and the nucleus is cardium. This nucleus is characterized by a large nucleus, central vesicular nucleus, achromatic, and it is with an evident nucleolus. And this reflects the active state of this cell. Also, if you look at the cytoplasm, you will see these granules, which are basophilic. These are the Nessel granules, which are the free ribosomes classified as focal ribosomes. These granules reflect the metabolic activity of the cell. The more they are present, the more it is an active cell. This is a very nice picture showing the lipofuscin pigment, which is one of the inclusions in the nerve cell. This pigment you see here is stained by golden brown color, and it takes a golden brown color. It's a normal color of it, and it represents the residue of undigested material by the lysosomes. Their amount increases with the advance of age, and they appear by this color in the nerve cell. So the more we are old, the more we have this golden brown pigment in the cytoplasm of our neurons. Here it is. This is a blood capillary, and all of these are the nerve cells, and this is the nucleus, and this is the nucleolus. So this is golden brown pigment is the lipofuscin. You may be asked, mention another site you know that has also this lipofuscin with the advance of age. The other side is the cardiac muscle. This is a very easy picture for a section, transverse section in the nerve trunk. We all know that the nerve trunk is formed of bundles of nerve fibers. And these nerve fibers are myelinated nerve fibers. It is supported by a connective tissue covering the whole nerve trunk called Perineurium. It is a normal or an ordinary connective tissue called Perineurium. And then we have another connective tissue surrounding the bundles here, there, and so on. This is called peri. Peri means around, حول, perineurium. And inside the bundles we have the myelinated nerve fibers, but they are not evident in this low power magnification. So inside these bundles we have the nerve fibers separated by the endoneurium, which is not evident in this picture. In this slide, you may be asked about the nature of the epineurium and the perineurium. The epineurium is this one is a dense connective tissue covering the whole nerve, and of course its function is the support. While the perineurium, this one around the bundles, this is formed of flat epithelium-like cells, flat epithelium-like cells that are joined with tight junctions, and these form a very important barrier. So a barrier is formed by the perineurium, very important to protect the nerve fibers. So the barrier is present in the perineurium as the cells which are flat cells like the epithelial cells, but they are joined with tight junctions, flat epithelial-like cells joined by tight junctions, forming a barrier to protect the nerve fibers. This is also a very nice picture demonstrating the nerve trunk. We have in this picture the perineurium forming a barrier to protect the nerve fibers. We have here in between the bundles, we have in this connective tissue, we have blood vessels. Also, we have here some fat cells. The bundle itself, when we magnify it slightly, it is formed of the nerve fibers. Nerve fibers, large numbers of nerve fibers, and separated by endoneurium, but it is not evident in this picture. The nerve fibers here appear as these circles having a white content inside, representing the dissolved myelin sheath, and you can see also nuclei representing nuclei of the Schwann cells. This is also another picture for the structure of the nerve trunk. Here he is magnifying the nerve fibers. In this picture you can diagnose that we have the nerve fiber like this, and in the center we have faintly stained axon, and the area here, this area here, it should be the place of the myelin sheath, but as it dissolves during preparation of the section in alcohol, we have only here just the framework which is called neurokeratin. Neurokeratin, it is the only thing present here, but it is also not stained, and this area was occupied by the myelin sheath. And in some of the fibers you can diagnose the nucleus of the Schwann cell. So if you have a label on the dot, this dot inside, this dot inside which is faintly stained, it is the axon, faintly stained. If you have a label for the white area, it is the site of the myelin sheath which dissolved during preparation of the section. If you are asked what is now present in this area, you can answer neurokeratin. And then if you have a label for the red cycle, it is the neurolemmal sheath. if you have a label for the nucleus on this red cycle, it is the nucleus of the Schwann cell. And of course, you know that the neurolemmal sheath is formed by the Schwann cell, and this is the nucleus of the Schwann cell. But in order not to make a mistake in your answer, it's better to give it this circle, to give it the label neurolemmal sheath or membrane, and this is the nucleus of the Schwann cell. Here we may be asked about the Schwann cell. What do you know about, or what are the features of the Schwann cells? The Schwann cells are one of the neuroglial cells that are present only in the peripheral nervous system around the axons. What is the origin of these Schwann cells? It is ectodermal in origin. What is the function? Is the production of the myelin sheath and the electric insulation and the regeneration. So this is a higher magnification for a part of the nerve bundle, showing here these circles. These circles are the nerve fibers. Inside the circle, we have a faintly stained area, which is the axon. And this area, which is white, it is the site of the dissolved myelin sheath. And this is the outline, the red outline here, it is the neurolemmal sheath, and this is the nucleus of the Schwann cell. If you have a label on the very narrow area in between the nerve fibers, this is the endoneurium. You may be asked, what is the nature of this endoneurium? The endoneurium is formed of reticular fibers. Reticular fibers formed by the Schwann cells. يعني الجوان cells here, which form the neurolemmal sheath, also forms the reticular fibers of the endoneurium. This is the light microscope picture for the nerve trunk, but here it is stained with osmic acid. The osmic acid dissolves in the myelin sheath, so that we can see here the myelin sheaths as these black circles. And the center of the circle is empty because it is the site of the axon, which is not stained. So here we have a nerve trunk formed of bundles. The surrounding connective tissue covering the whole bundles is called the Perineurium, and the connective tissue surrounding the bundles itself is called the perineurium. Connective tissue or site of connective tissue here in between the nerve fibers, it is the endoneurium. Here it is not stained as it is not stained by osmic acid, but its site here can be seen, and it takes the stain non-specifically. Sometimes you can see these black circles, but it is non-specific stain. It is enforced to take some color by the stain, but not non-specifically stained. Here we have the most important thing is the nerve fibers. The center here, it is the site of the axon, not stained, and the black circles demonstrate the myelin sheath stained black with osmic acid. Again, this is a nerve trunk. Bundles of the nerve trunks can be seen, and it is very nice picture. You can see non-specific staining of the perineurium, and also here we have the site of the endoneurium, and the most important things are these nerve fibers showing the myelin sheaths here in this black coloration. This slide also demonstrates the nerve bundle in a nerve trunk stained with osmic acid. And here we have very important things. We have this is the myelin present in the myelinated nerve fiber, and the white area here represents the site of unstained axon, and this is the perineurium. And these areas here around the nerve fibers, this is the endoneurium, which is formed of reticular fibers made by Schwann cells. So these are the myelinated axons, and this is the endoneurium, and this is the perineurium. In this slide, you may be asked about very important things about the myelin sheath. First of all, it is formed by, number one, formed by what? Formed by the Schwann cells. How? By making several turns around the axon. So this is as regards the myelin sheath in the peripheral nervous system. But in the central nervous system, it is formed by other cell, which is the oligodendroglium. The Schwann cell, when form the myelin, they make a wrap, several rounded wraps around one segment in one axon. Very important, because the oligodendroglia make another action. It make wraps around one segment of many axons, but here it form several turns around one segment only of one axon. So the myelin sheath is interrupted at the nodes of Ranvier. Nodes of Ranvier. Why we have these nodes? Because the Schwann cells, during formation of the myelin sheath in the peripheral nervous system, form several wraps around one segment of a single axon. So the segments are separated from each other, and each segment is formed by one Schwann cell. You may be asked also about the functions. The functions of the myelin sheath are very easy. The myelin sheath, form an insulator around this segment so that it enhances the speed of nerve impulse through the saltatory conduction. This is a very important picture for the spinal ganglion. How can you diagnose the spinal ganglion? From the following points. First of all, the cells here are present in groups or in rows. This is very important. This is a group, and these are two rows. And this is very peculiar to the spinal ganglion, differentiating it from the sympathetic, in which the cells are scattered in the whole ganglion. After that, the cells here appear to be rounded because they are unipolar cells. They have one process that divides into two branches, one long branch to bring the sensations from the skin, and one short to carry this sensation to the spinal cord. Also here the cells are of different sizes. We have large size, we have intermediate size, and we have a small size. Each size is responsible for a certain sensation. Also here these are the most important features. If you are asked, mention one or two features that help the diagnosis: the arrangement, first of all, and number two, the cells are variable in size. They are very evident in the picture. Also, you can add, if you are asked for three reasons, you can add that the cells are more or less rounded, meaning that they are unipolar cells. So the arrangement and the size and the shape. After that, you may have a label for this one. This is the blood vessel, and all we know that the blood vessels are very minimal in the spinal ganglion. If you are asked, is there a synapse in this ganglion? No, no synapse. Why no synapse? Because the cells here are unipolar, and the cell itself brings the sensation by its own and carry this sensation to the central nervous system. So it does not depend on a synapse with another neuron. So this is no synapse in this ganglion. You may be asked, if you are diagnosed that it's spinal ganglion, you may be asked what the type of these structures. These structures are the axons of these nerve cells. These axons are myelinated. So these are myelinated nerve fibers present in between these bodies of nerve cells. This is a picture for the spinal ganglion, and here we have a covering connective tissue capsule. Actually, this capsule should be thicker than that of the sympathetic ganglion, and this should be taken in your consideration during drawing the ganglion. Here we have the nerve cells, and inside the cell you can see this pale structure with a dot inside. This is the nucleus, achromatic nucleus with a prominent nucleolus. And then you will notice a gap around the cell, then a circle of supportive neuroglial cells called satellite cells, described before. Here we have the nerve fibers, red and blue lines. These represent or pale red and dark red. These represent the myelinated nerve fibers. So we have this circle of satellite cells. This is the nerve cell, and this is the nucleus, and the dot here is the nucleolus. We can magnify it more. These are the satellite cells, low-cuboidal cells, neuroglial cells in the peripheral nervous system around the cells of the ganglia, and this is the nerve cell. Always there is a gap between the servant and the queen. We have this gap. And then we have this nucleus with achromatic appearance and the nucleolus, and this is the cytoplasmic granular, as you see, representing the Nessel granules. This is another picture for the ganglion, but now it is stained by silver. The silver in the nerve cell will stain the Golgi apparatus and the neurofibrils by the brown color. Here you can diagnose that we have a covering capsule, we have group, row. So this is the spinal ganglion separated by myelinated nerve fibers. So this is a spinal ganglion stained by silver. A picture for another ganglion. It is the spinal ganglion and it is stained by silver. Why it is a spinal? Because we have a big group, then a space formed of myelinated nerve fibers, then a row, space filled with the myelinated nerve fibers, then a row, and so on. They are not scattered. We have an areas occupied by these myelinated nerve fibers, evident areas separating these groups or rows. So it is the spinal. What else? The cells here are not uniform in size. We have big size, we have small size, we have intermediate size, and each size is responsible for a certain sensation to transmit to the central nervous system. Of course, the brown coloration suggests why there is brown coloration, because the Golgi apparatus, which is well developed and fill the cytoplasm of this neuron in a position around the nucleus, is stained by silver. The Golgi apparatus here is well developed and it is present around in the form of a network around the nucleus, is stained brown by the silver. Why we have brown coloration here in the nerve fibers? Because in the nerve fibers we have the neurofilaments, which are stained brown by silver. This is another picture showing the spinal ganglion stained by silver. How did you diagnose that it is a spinal ganglion? Because of the two things: the arrangement of the cells in the form of groups or rows. Number two, the variation in the size. We have large size, we have a small size, we have an intermediate or moderate intermediate size. Here we have also the brown coloration around the nucleus. Why? Because it stains the silver stains the Golgi apparatus, which is well developed network around the nucleus. Why we have brown coloration for these nerve fibers? The brown coloration here due to the staining of the neurofibrils 2 micrometer in diameter in the nerve fibers by the silver. And as I said before, these neurofibrils are nothing but stuck neurofilaments together to form a larger bundle due to the process of the fixation used during preparation of the slide. This picture is a very nice picture for the sympathetic ganglion. How can you diagnose it is a sympathetic? By the following. First of all, the cells are scattered in the ganglion. Second, the cells are uniform in size. Third, the cells are small in size. And finally, the cells are not unipolar. They are showing processes. They are angular in shape, denoting that they are multipolar stellate. In this picture, you can notice this capsule, which of course, compared to the spinal, it is thinner than that of the spinal ganglion. Also, you can notice here the fibers. The fibers here are of the unmyelinated nerve fibers. These fibers are unmyelinated nerve fibers. Here, another picture showing many important data. Here we have the nerve cell, and this is the nucleus, and this is the nucleolus. And this cell should be in a ganglion because it is surrounded by these low cuboidal cells called satellite cells, so it is in a ganglion. Here we can see also in this cell this golden brown color of lipofuscin granules or lipofuscin pigment discussed before. Okay, here you can see that we have also in between these nerve cells, we have also these red and blue representing the nerve fibers. In this picture, you can see that we have many blood vessels. Here, this is one blood capillary with blood, and this is another one. Also, the cells appear to be scattered in the section. The cells are not so rounded, they are angular, so most probably it is a sympathetic ganglion. This is another picture for a ganglion stained by silver, but now it is not the spinal ganglion. Why? Because we have the cells scattered in the sections, and the cells are all small in size, almost uniform in size and small. So this scattered appearance and uniform small size represent the sympathetic ganglion. What is the type of the neuron here? It is a multipolar stellate neuron. Do you have a synapse in this ganglion? Yes, there is a synapse. We have a preganglionic and postganglionic. What the type of the nerve fibers here? We have here the postganglionic fibers are unmyelinated nerve fibers. This is a picture for the ganglion. Why? Because we have a large collection of nerve cells. Each nerve cell is surrounded by an empty white space. These cells are stained by silver. So I should diagnose, is it a spinal or sympathetic ganglion? As the cells are almost of the same size, as the cells are angular in shape, not rounded, so they are multipolar stellate, as the cells are scattered in the section, so these are the three criteria by which I can make my diagnosis that it is a sympathetic ganglion stained by the silver. Why the cytoplasm is stained brown? Due to the well-developed Golgi apparatus. Why the nerve fibers here? The nerve fibers here are here appear brown due to the presence of the neurofibers that stain brown also by silver. This is the sympathetic ganglion also stained by silver, but of a higher magnification. You can see here that the cells have an achromatic nucleus. The cytoplasm is brown due to the Golgi apparatus well developed. Here the nerve fibers due to the neurofibers in the nerve fibers stained brown by silver. Why it is sympathetic? Because the cells are uniform in size. They are scattered in the section. They are most of them show angulation, meaning they are angle. This means that they are multipolar. Also, they have uniform size. Almost all of them are of the same size. No marked difference can be seen in between them. Let's start by the first slide in the trial exam. The wavy arrow points to the satellite cells. The arrow points to the pericaryon of the unipolar nerve cell because it is almost rounded with no angulation, with no processes, so it should be the unipolar, and the unipolar is a very important point in your diagnosis. So it is the pericaryon of the unipolar nerve cell. The arrow head points to these fibers. These fibers are present in the spinal ganglion. As you see, the cells here are separated by fibers, and they are present in group separated by fibers. So these and they are unipolar. So this is the spinal. So these fibers are the myelinated nerve fibers. Identify the organ sympathetic ganglion. Why? Because the cells are uniform in size, scattered in the section. The arrow points to. هنا الارو بيشاور على البراون structure اللي عندي ده. So this is the Golgi apparatus in the pericardium. After that, we have, if you have a label for these fibers, this is the unmyelinated nerve fibers. What is the stain? The stain is silver. The wavy arrow points to the nucleus of the Schwann cell. The arrow points to the axon or axis cylinder. And if you are asked about, and here we have the arrowhead, it is pointing for the neurolemma or neurolemmal sheath. If you have an arrow in this white area, it is the site of dissolved myelin sheath. Also, if you are asked what is this structure, it is the nerve bundle in a nerve trunk. Identify one. One is all this structure. This is the label one, and it points for these dots. So all these dots means one. So this is the pericaryon of the neuron. Number two, it is the nucleus of the neuron. Number three, they are the satellite cells. If you are asked what the type of this neuron, it is of course unipolar nerve cell. The arrow here, this is the arrow, points to a cell called neuroglial cell. Its most common type is the astrocyte. The arrowhead points to the neuron, which is multipolar stellate. This is the pericaryon of the multipolar stellate nerve cell. The arrows point to the, here, what is this structure? This is the perineurium. Peri as it surrounds the bundle. If you are asked about its structure, it is formed of flat epithelial-like cells joined by tight junction, and it acts as a barrier. What is the function? Red circle surrounds the bundle of nerve fibers in a nerve trunk. Arrowhead here points to the myelin sheath, stained of course black by osmic acid.